I'm going to talk a little bit about how interested in the microenvironment and particularly on the, going back, particularly on the glycocalyx of the cells. I don't have a pointer. No pointer. Non tem bateria.
I get one for you.
Sorry?
I get one.
Please. I will need to point, yes. So we are very interested in the glycocalyx of the cells, and we have been studying these molecules, which includes obviously glycosphingolipids, glycosaminoglycans, and glycoproteins. And within glycoproteins, we have two main types. You have the O-glycans. Sorry?
Is it working? No.
No?
We try to fix it.
Okay. So we have these O-glycans and N-glycans on glycoproteins. And I cannot move. Yes, thank you. Two problems. Now it does. So on these glycoproteins, we have these major types of O-glycans and N-glycans that can occur in every eukaryotic cell. So very briefly, on N-glycosylation, which will be the topic of the work that I'm going to present, is a very controlled pattern of, in the cell of biosynthesis of these N-glycans, which occurs in the reticulum endoplasmatic of the cell by the action of different enzymes. And this forms a precursor structure that is transferring block to the aspargine of the protein that is being synthesized, either for a secretor protein or a transmembrane protein. Obviously, a protein can have more than one site of N-glycosylation, so we will have different sites of N-glycosylation. Then the protein, as you know, it moves from the ER to the Golgi apparatus, and there it will undergo major alterations by family of glycosyltransferases, which in the human genome, there are nearly 200 genes that could modify and form different glycans, different N-glycan forms. In fact, this is highly controlled according to the differentiation of the cell in tissue and cell-specific pattern of glycosylation that we can see, for example, as in this example, in the gastric mucosa, where you have a specific biosynthesis of certain glycans following the foveolar epithelium superficial in the gastric mucosa and other glycans biosynthesis in the deep glands of the gastric mucosa. This is highly controlled in normal conditions, in physiological conditions. And in what we know in cancer is that several mechanisms that I don't have time to go into detail lead to the biosynthesis of certain structures aberrantly expressed, such as the shorter truncated or CLOTN antigen, the Lewis antigens, for example, the cellular leucine, most clinicians know because this is the tetrasaccharide that is detected by the CA19 serological assay used in the clinics, and also this alpha-2,6 cellulation of N-glycans. So the model that we have studied and I'm going to present today is gastric cancer, which is a highly incident tumor in the world. Not only is the sixth incidence, but also high mortality. And as you all know, it's a quite heterogeneous tumor, and basically molecularly, there are several subtypes of gastric carcinomas. And the therapeutic approach follows several strategies depending on the progression of the disease. And in advanced tumors, the tumors that are, some of them are positive for the ERB2, and this can be, it has an approved treatment using trastuzumab. So, and I don't need to mention, but as you know, trastuzumab is a monoclonal antibody humanized to target this receptor, and it acts as disruption the ligand-independent heterodimers that it can be formed in the tumor cell. The receptor internalization and degradation inhibits signaling, intercellular signaling, and also induce ADCC. The interest in this target at that time, some years ago, it was that because we observed that this protein had different sites of N-glycosylation in extracellular domains, including in domain 4, which is the binding domain of trastuzumab. And also because we clearly demonstrate that at that time that when we remove the N-glycans using an established enzyme that removes N-glycans, you can see a clear shift in the band of the protein. And moreover, when using different lectins, including this alpha-2-6 detecting lectin, obviously if you remove the N-glycan, you don't detect the acylation. So highlighting that this protein was actually acylated with that in these N-glycans. So we decided also to see if this was really occurring in tumor cells and actually in tumor tissues from patients. So we study expression of ERB2, of course, in gastric adenocarcinomas, the SNA, the alpha-2-6 acylation, which is detected in tumor cells, but also in other stromal cells. And when we study the close proximity between these two targets in the tumor, using this strategy which detects on one side the glycan and on the other side the protein, we see that some tumors were giving clearly signal and others did not. And actually when we observed in this cohort that this signaling, PLA signaling for alpha-2-6 acylation and protein was also discriminating between different types of tumors in the same stage, but with different survival behavior. So we decided to go a little bit in depth in this, and we target the enzyme, the STC-GAL1 enzyme, which is the one responsible for the alpha-2-6 acylation in N-glycans. And here we just describes the clones, independent clones, lacking the enzyme, but also lacking the product of the enzyme, as detected by this lectin, also by this FACS analysis, and decided to go in depth in this and characterize the glycosylation of ERB2. So we immunoprecipitated the protein from these clones and perform a different glycosylation studies. One using glycomic analysis, in which you remove the N-glycan of the protein and label chemically by click chemistry and analyze and quantify the glycan present in this protein. And then we also, in a complementary study with a glycoproteomic approach, in which we digest the protein with a proteolytic enzyme, and then analyze the peptides containing the different glycoproteins by LC-MS mass spectrometry. In a very summarized way, we observed that we had a reduction of alpha-2-6 sialic acid, also called neuraminic acid, in the clones compared with the control. We have a slight increase in alpha-2-3, and we have a major increase in terminal fucosylation, because obviously the final product of this biosynthetic pathway, when you remove one target, you make available the different substrates for the action of other glycosyltransferases. In detail analysis, we observed that there was confirming the increased alpha-2-3 acylation in the clones compared with some of the species, and also, as I mentioned, an increase in fucosylated structures consistently in the different N-glycoforms present in ERB2. And as expected, a major reduction of sialylated alpha-2-6 acylation because of the knockout of the enzyme. If we go into detail, the glycoproteomic analysis further provided information of the specific N-glycan form in each N-glycoside of the protein. So basically, this shows an example of the aspargine 530 of the RB2, which in this case, this glycopeptide containing this glycoform by sequential fragmentation of this precursor ion, we can have exactly the structure on that glycopeptide. And so we can define exactly which glycoforms were present there. And summarizing a lot of data, we could actually define what are the glycomap present in RB2. And was very interesting to find that many of these alterations were occurring in the trastuzumab binding domain, domain 4. So when we analyze the behavior of these clones compared with the controls cell, we see a clear reduction it makes the knockout cells the clones are more sensitive to the induced cytotoxicity by the treatment with the trastuzumab antibody, and also induce higher level of membrane anchor RB2 indicating a prolonged stabilization of the RB2 trastuzumab complex in the membrane of the cell. Furthermore, we observed that there was a clearly that the knockouts show that the cells were more makes more sensitive to the RB2-driven, in this RB2-driven gastric cancer cells, to the trastuzumab treatment, leading to downregulation of the RB2, EGFR, dimerization, dimerization as observed here. In fact, the clones show reduction sensitivity. And also, we observed that there was an induction of extended time, extended half-life of the RB2 receptor in the clones when compared with the control. So in summary of all this data and some that I didn't have time to show, we observed that the gastric cancer cell display specific glycoforms on RB2 show different response to therapeutic antibody-induced cytotoxicity. And in fact, the increased alpha-6 cellulation of RB2 confers increased resistance to therapy. And the take-home message at this stage is that then glycosylation glycoforms of RB2 impacts the cell, not only the cellular biology of the receptor in gastric cancer cells, but also the glycoproteomic information of RB2 show that the modification of the cellulation and this fucosylation that can occur on RB2 in cancer cells can change the activity and the response to the trastuzumab. Cancer cell displaying specific glycoforms of RB2 showing different response to the therapeutic antibody. So I could not finish without highlighting that this, on one side, corroborates previous studies demonstrating that tyrosine kinase receptors can be dramatically influenced by the function by glycosylation, not only of themselves, but also on glycosaminoglycans and proteoglycans, but also highlighting the function of that some of other proteins can be playing in this heterogeneity of the tumor in the interaction with extracellular matrix, but also in distant sites of metastasization due to the, because some of these carbohydrate structures are the actual ligands of selectins in endothelial cells. And moreover, in the context of the tumor microenvironment, that nowadays it is well known that some of these carbohydrates are particularly expressed in the tumor, as I mentioned, but also are read by the lectins of the immune system and also by the by the endothelial cells, as I highlighted here. So I think I've run out of time. So with that, I just thank all the members of the team, particularly Henrique Duarte, who was mostly responsible for the work I presented today. I would like to thank the collaborators and also highlight the funding, the support, and mention the Porto Comprehensive Cancer Center Raquel Saruca, which has the name of a colleague that we lost this year. Thank you very much.